Welcome everyone to the October AIRR-C seminar. Today we have two speakers, Scott Boyd from Stanford University and Suhas Sureshandra from the University of California, Irvine. First, we have Scott Boyd, who's going to talk about human B-cell responses. And so to briefly introduce Scott, he's a physician scientist, Stanford professor in food allergy and immunology, co-director of the Sean Ann Parker Center for Allergy and Asthma Research, and professor of pathology in the Department of Pathology at Stanford University. And his lab uses DNA sequencing, single cell experiments, and other tools to study the B-cell response in infection, vaccination, as well as immune disorders such as food allergy and immunodeficiency. He was also the recipient in 2019 of the Presidential Early Career Award for Scientists and Engineers and was elected to the American Society for Clinical Investigation in 2020. So I'm very much looking forward, Scott, to your talk. The floor is yours.
Thanks very much, Victor, and thank you to the organizers. I think it's really great that you guys are having this seminar series. So really happy to be able to present and to see many friends and familiar faces and names in the group that's attending here. Okay, so I'll get started. Let's see. So this stuff I'm going to talk about. Yeah, the title is Determinants of Antibody Breadth After Infection or Vaccination. And these are some of the main players from the lab who've done the work that I'm going to talk about. And I'll get right into it and mention people as we go along. So let's see. Yeah, so just to set the context, like many labs, we're interested in a lot of the factors that contribute to the humoral immune protection or humoral immune pathology that develops in people. And some of the main questions we've been trying to figure out, which was really sort of accelerated during the SARS-CoV-2 pandemic, include things like what happens when your immune system responds to antigens in different contexts, like an infection with a virus compared to vaccination with different kinds of modalities. And in particular, one of the interesting characteristics is, what determines the breadth of antigen variant recognition by B cells. That's something I'll touch on a little bit today. A lot of that is tied into aspects about, well, how does your immune memory or your prior exposures, how do they help to sort of shape or alter the way you respond to new antigens that might have some similarity to what you've seen before, or in some cases be completely new. And then part of this is also trying to look in tissues that have been less accessible to investigation in humans, like the lymph nodes, spleen, different mucosal immune sites, for example, compared to the blood. That's the basis of most human immunology work. And in addition, we're quite interested in how the immune system's responses are altered in circumstances where there's changes in some of the other cells that support B cells or that have other roles in the immune system, and also in sort of characteristic conditions that have B cell problems or altered immunity. So the first, I'm basically going to talk about some SARS-CoV-2 related data and then talk about some influenza work. And with the SARS-CoV-2 topic, we had, like many labs, been very busy during the pandemic, trying to understand the antibody responses and initially, of course, to the infections by the new virus, but also vaccinations as they came along. And we had made some observations serologically fairly early on that we found puzzling. One of them was that if we looked at the serological response to SARS-CoV-2 in vaccinees who had received the Pfizer mRNA vaccine, the first version of it that came out that had the, close to the original SARS-CoV-2 variant in it, compared to individuals who had been infected in the first months of the pandemic and either had sort of milder infection in orange or severe infection in red. What we saw is if you looked at the titers of antibodies in each of those contexts, for one thing, the mRNA vaccines gave really good antibody responses, quite high titers, targeting the spike, the RBD, not the nucleocapsid, of course, which isn't encoded by the mRNA vaccine. But so the titer response was good, and we saw lower antibody responses in people who had less severe disease, But we also noticed, as many did, that there was already a sort of early drop off, even within a few months of the antibody titers, and that continues basically until they become relatively low within 6 months after getting the vaccination. Um, there was another aspect to this, which is the breadth of the serological response. So here we're measuring the polyclonal antibody responses in people after those fairly simple exposures, like infected early in the pandemic or first round of mRNA vaccines in people who had not been infected to that point. And one thing is that we noticed is if we did immunoassays for different SARS-CoV-2 variants that showed up later in the pandemic, and we compared the relative binding we saw to the antigens that were of these different variants compared to the original SARS-CoV-2, the Wuhan-like viruses or the Seattle variant. What we found is that if you plot those as a ratio, in terms of whether each variant is bound better or worse than the original SARS-CoV-2 variant, they all were basically bound less well than the original antigen, which makes sense because they were infected with the original virus um, or had the mRNA vaccine that encoded a very similar protein. But we noticed that there was quite a difference between patients and vaccinees in this proportion, like how well they bound their variants compared to the actual original SARS-CoV-2 spike. And this was done with measurements with the MSD, so mesoscale discovery immunoassays, where you have all the antigens deposited, like in little patches on the bottom of the wells of a 96 well plate format assay with electrochemiluminescence development. And so it's a really well controlled assay, internally controlled within each well. You have each of the antigens, and you measure the light emitted from each patch. So we thought this comparison was one of the more exact ones about how much antibody was bound to each of the variants compared to the original SARS-CoV-2. And so what we saw is that the patients initially had um, you know a lot less breadth of their antibody binding compared to the vaccinees, but that over time that decreased. So by the time we got to 3 months, they were actually much closer to each other. But that for some reason, getting the mRNA vaccine with the modified RNA encoding the spike was giving a relatively better breadth, so lower on this y-axis than the infection with the virus. And that to us was a bit surprising because we thought maybe the overall immune stimulation of having the viral infection, all the other kinds of innate activation that go on in the context of a viral infection would have given a more, a bigger immune response. But it seemed that there was this difference in the antibody specificities that were elicited. So with the serological measurement, we then tried to, and this was in the context of developing these kind of assays for other contexts as well. We were trying to, improve the ways that we could actually measure for at the level of individual B cells, individual B cell clones, measure the specificity of individual cells in bulk or on large scale. And so for these kind of experiments, we made use of some of the single cell transcriptome platforms. In this case, it was the 10x platform we used predominantly. And people had developed in, some other labs like had been starting to use DNA tagged surface antibodies or DNA tagged antigen complexes as a way to try to measure the specificity of B cells like you would in a flow cytometry fluorescent tagged antigen complex sort of experiment. So here we have the samples coming from patients, and some of them are blood sample PBMCs. In other cases, lymphoid organ samples, like from deceased organ donors, where we're able to get the fresh spleen and lymph node and blood samples from them. So samples containing B cells. And then here we've had panels of viral antigens purified, made in a biotinylated form, either from the point where they're expressed using a beer tag or a way you can attach biotin at a particular site very specifically, or by later biotinylating just chemically as you can do. So we make these complexes of streptavidin where each streptavidin has a particular antigen in a pure form attached to it by the biotin. And each complex has a fluorophore and it also has a DNA barcode that tells you what the antigen is essentially. For each of the antigens that we use in these panels, we made two versions with two different fluorophores, and they have the distinct DNA tags. So that way we can do sorting based on the double positivity and get rid of B cells that bind only the dye, for example, or other parts of the complex that are not specific to the antigen in each case. So we made panels of these antigens, a bunch of them, mix them with the B cells in the sample, sort using a flow sorter, the B cells that bind the, any antigen in the pool, and then put them into the 10x or other single cell platform to get gene expression data, B cell receptor data, and the antigen binding to each B cell data based on the DNA tags that we can read out in the experiment. So doing this, we've analyzed over 5,000 antigen-specific B cells in various different experiments so far. But so it's actually, and if you look at the CoV-AbDab database of defined specific sequences, the last time I looked, there were maybe 12,000 total or something. So in the course of like a few weeks of experimentation, we can basically identify specific cells on a similar scale to that. So in the data I'm going to show, it's based on a subset of the patients who we've studied overall. So in various ways. So 28 COVID-19 patients, they varied in disease severity, but so far we haven't found a major difference in the specificities I'm going to talk about based on the severity in these individuals. They were all infected in the first few months of 2020 in California. So they're basically exposed to the early versions of the virus. And we also compared that to 17 individuals who'd received the first type of Pfizer-BioNTech mRNA vaccine, encoding the sort of Seattle type virus spike. For these, we had a time course of samples. Same thing with the COVID patients, and the time points were pretty well matched. So this is work that was done a lot. Katrina Rotkin, I want to call out because she did a lot of this work. She's now at the Swiss Tropical and Public Health Institute in Basel, and our various collaborators that are locally here. So these are the antigen panels that we used in these experiments for SARS-CoV-2 that I'll show. So the first antigen panel, we basically had the full spike, NTD domain, S2 domain, nucleocapsid, and then we had a bunch of RBD variants. So in this case, it was 8 different RBDs. This was pre-Omicron when we were setting this up, so it was really the ones that were leading up to Omicron, basically. In this second antigen panel, we extended that list of RBDs considerably, and a lot of these are single point mutations in the RBD, so it's almost like, it approaches like an epitope mapping kind of scheme in a way. We also included one non-SARS-CoV-2 beta coronavirus, or the OC43, just the RBD. So you can see that with this kind of approach, right, if you get the signal from a particular B cell, you should be able to tell, both does it bind the antigen, some part of the spike in terms of which domains. Having a separate, for anybody doing this kind of work, I would say having independent kind of proteins that are not at all related to the main ones you might be interested in is really useful for helping to set the thresholds for what's really positive binding or not. This is something that a bunch of labs are tackling, like where do you draw the thresholds for positive binding with these DNA tagged antigen sort of experiments. So the nucleocapsid is useful in that regard in this experiment. And then all these RBDs. And the question will come up, which is basically how many variants of a similar protein can you have in a panel like this before you start to get competition that would actually decrease your signal and give you false negatives if you have many things that are related to each other? Because, apart from the places where they have mutations, other epitopes that B cells might want to bind to, you may have quite a bit higher concentration of the antigens with a particular epitope. And so the B cells are basically exposed to a higher dose of that, but they're each having their own DNA tag. So the amount of signal for each one becomes potentially less if the B cells becoming saturated for binding. So just to show you how that looks in terms of the actual sorting part of the experiments, the Pfizer vaccinees received the 2-dose regimen. So day 0, day 21. We don't see much in the way of specific B cells. So this is the spike with APC fluorophore spike versus brilliant violet. And in the initial days after the first dose, you don't see too many specific B cells in this periphery. That's pretty typical for primary responses of all kinds. And by 7 days after the second dose, we do see a nice population that looks quite specific based on the 2 fluorophore dye sorting. And they sort of diminish in their frequency over the next couple months. This is just 2 different people showing their time course. If we look at the frequencies plotted, it's pretty typical. This is how most people respond to the vaccine. In patients, you see a different pattern of time course. So if you look at similar times relative to the onset of symptoms, what we see is more a pattern of progressive increase of the frequency of the specific B cells through at least the first 3 months. And we think that's a combination of things, potentially the gaining of avidity or, the better affinity improvement of the B cells in the infection. But also there not being such a sharp, initial exposure to antigen that happens with the mRNA vaccine. If we look at the nucleocapsid specific B cells, we see not much in the vaccinees as you'd expect, and a similar pattern to the spike reactivity in the patients. So it looked like the, B cell sorting aspect was working okay. Then now if we look at the single cell transcriptome data from these cells, um, and this is work that a number of people worked on in the lab, but particularly Fan Yang and show you the yeah, basically Oliver did a lot of the sorting and setting up of these panels, a ton of really high quality wet lab work in this. Fan Yang is doing a bunch of the data analysis initially for the SARS-CoV stuff, followed by Shanti Kodagiri, who was a Fulbright fellow in the lab, who's done an amazing job with some of this next stuff. Katalina was involved in some of the SARS-CoV-2, the main SARS-CoV-2 serology, as well as a bunch of the flu work that I'll talk about that Brandon was also doing. And Taylor is pitched in for analysis of a bunch of these. Artem has recently taken on the flu projects with a big enthusiasm. So just to give full credit where it's due, because these are the people that really did the work. Okay, so the phenotypes that we saw, breaking it down to memory B cells, some naive B cells came through the sorting. So we, depending on the day, we either included some naive B cells or not. So don't interpret much from the proportions of these. But we thought it would be interesting also to see if we detect any binding in the naive pool of some of these antigens. And then these are plasmablasts or other antibody secreting cell phenotypes. And, in full disclosure, in the course of doing the long day of getting the samples, thawing them, labeling them, sorting them, etc., even though we sort for viability, by the time we actually get to the single cell transcriptome data, some cells have started to lose viability. So that's this population here, based on the mitochondrial DNA and ribosomal RNA counts. So here she sees some main clusters. Within them, we've done subclustering, which I'll get into. But you can see here, these are the atypical memory B cells that have CD11C, Tbet in some different proportions. And non-switch memory B cells are largely here. This is a population I'll mention more in a minute, but they have an activated phenotype. And then these are the main class switch memory B cells over this data. This is combining the data from the vaccines in patients. And one nice aspect about this was we were able to do the sorting kind of nicely combined with infected and vaccinated patients. So we're able to avoid batch effects based on the sorting and the other, experimental day kind of details. Here are the antigen-specific cells. So we also sorted some that were non-antigen binding based on the flow cytometry. And the colored dots tell you what antigen each B cell was detected as binding. So there's a lot of overplotting here, but you can see that there's a real enrichment of specific cells in some of the B cell populations. We also detect some binding to occasional ASCs or plasmablasts, and a few naive B cells also picked up the signal. Sadly, some of the cells that were showing low viability also found the antigens. We think that a bunch of these are real signals that were just the cells dying in the course of the experiment based on their clonal relationships to some of the more viable cells. We can suspect that that's actually a real binding signal. Okay, so if we look at the phenotypes, here the gene expression, I won't dwell on this too much, but they're all fairly typical ones, CD20 and 19. The ones distinguishing the atypical cells are here. This is Tbet, basically the CD11C. And some of these interesting genes, like these FC receptor like proteins, these seem to be good markers, at least at the transcriptome level, for different subpopulations. Here are the plasmablast kind of markers, XBP1 and high CD38, like you'd expect. And there were a significant number of proliferating cells in the time points that we saw for these data. So what are the differences between infection and vaccination? One thing that we noticed right away is that if you look at the infected individuals, there's this population that you really don't see in the vaccinees, even though the time points are pretty similar for the blood samples that were collected. So what are they? We also noticed that there were a few people that had prior infection when they were mRNA vaccinated. So we captured the time points from their mRNA vaccine response, but despite their prior infection, we still don't see a population like this one in those people. So when we split it out by the infected at the different time points, this shows more of the, less combined data. It seems like that's true at all the time points that we looked at as well. There's this pretty significant population, including some specific cells. So these have a gene expression signature of what we've been calling cytokine activated B cells or AP1 pathway B cells. So they have a bunch of genes that you see in B cells that are responding to a variety of different cytokines in vitro experiments. So they really show a sort of gene expression characteristic of activated cells, including some of the obvious well-known markers like CD69, but a bunch of others that are part of the response pathways for exogenous cytokine signaling. So these seem to be something that's really characteristic of the infection, not the vaccination. Um, but were they the main place where the antigen-specific cells were localized? No, not really. There are some antigen-specific cells, but they're predominantly a lot of them didn't bind the antigens, at least that we included in our panel. So they may be responding to other things in the context of the infection, like antigens that we don't represent with these purified proteins, potentially ones that might also be reflecting things like, maybe damaged cells or other features of the infection. When we look at the antigen-specific cells, they were enriched in two other populations. One of them was the atypical memory B cells that I talked about briefly. The other was a subpopulation of the class switch memory. So these we'll go into more depth about now. Um, so there was this atypical B cells, as I mentioned, this subcluster of 15. Those have all the markers you'd expect, like, CD11C, TBET, NKG7 is up in them as well. A bunch of other characteristic genes. So these are pretty uncontroversial. They have high CD20 compared to some other cell types. This Z2 transcription factor has been implicated in the differentiation of this particular subset. So that's pretty clear. These have been tied to being involved in, um, extrafollicular B cell responses. And as I'll show you, like, I think some of the data that come out of these experiments help to support that. I've always been a little, you know, um, thinking that we didn't really have the definite data from humans to indicate that these really don't go through the germinal center, or not much, but that's something that I'll talk about. This cluster 13 of the antigen specific cells or enriched in them were interesting as well because they had a bunch of genes that we thought were pretty interesting that, for example, included IL-4 receptor, relatively high CD38, IL-21 receptor, and even AID, even though these are memory B cells, as well as Ki67 being relatively enriched in them compared to the rest of the class switch memory. So these we thought really looked like hallmarks of a cell that may have recently been in the germinal center, right? Especially the AID, but also these other receptors for the cytokines that are, notably present in germinal centers. So our current interpretation is that these particular class switch memory B cells, they may represent recent emigres from the germinal center that are still expressing some of the genes that were upregulated there. And they seem to be quite enriched for antigen specificity. That sort of makes sense in the context of the time points we're looking at for infection and vaccination. We saw them in both infection and vaccination, so they weren't, distinctly characteristic of one like these activated type were in the infected individuals. So then now we can get into the antigen specificities and what you learn from the different patterns of binding. So these are the DNA tag counts that we get for different B cells for example, the RBD for alpha compared to the Wuhan or close to wild type virus. So alpha, as you know, is not a big escape variant, just had a single change that wasn't very, deleterious to binding for most B cells. And so you see a really nice correlation of binding. The counts are fairly robust. It looks really like a flow cytometry kind of plot. If we look at variants that are more, escapey or basically have effect epitopes that more B cells are involved in binding, then we start to see populations that are negative for that variant compared to the original Wuhan. And when we look at the beta variant, we saw it even more so. In more recent experiments, if we look at Omicron variants, you see that to a greater extent yet in these early kind of exposures. We also find with the Delta that came along later a similar kind of pattern. So that's pretty clear. I should have mentioned the serological data we published in a paper that was in, I think, 2022 or so. That covers a lot of this if you want to see more details like of IgA binding and IgM and all that. Okay, so this is a table made from about 1,000 like a heat map, basically, from 1,000 peripheral blood B cells. Each little row is a cell, and each column is showing how much binding we detected by the DNA tag counts for the different antigens that were in the panel. So this is not showing every single antigen, but it's basically some of the main ones you'd be curious about. So notably distinct, here are all the nucleocapsid binding B cells. There's about 1,000, I think, in this table overall. Um, and so it's nice that these seem to be quite distinct from the various spike binding B cells. So these are the ones that bound the intact full spike from the Wuhan, all the ones here. And within them, we see subpopulations, as you might expect. Here are a bunch that also bind the NTD domain by itself. Here are the ones that bind different versions of RBD in different combinations. So you'll see, for example, here's the RBD Wuhan that was in the spike. These two should be, correlated quite well. Similarly, the alpha variant, which was very similar to the Wuhan original RBD, looks similar. The beta variant shows relatively less B cell binding, gamma less, delta less. Some other ones that are relatively more innocuous as single mutations, like the 417N, show a better correlation with the Wuhan binding, and other ones that are like the E484K, which is a key residue that's mutated in the beta and gamma variants, and some others. It shows relative loss of binding, but is better than the more complicated other variants that have more mutations. And cross-reactive ones for OC43, quite a low number that we detected in this panel. So it suggests that there's not a huge cross-reactivity, at least with this particular RBD. One other thing that's important to note is, the devil is in the details of these antigens. So when we look at B cells that bind the S2 domain, if you have S2 by itself, in most of the ways that those constructs are made, we think that it's not necessarily fully intact and trimerized all the time. It may sort of open or make other epitopes accessible that you don't see in the spike trimer. These trimers were quite stable and solid. So we actually get some B cells that bind S2 that don't seem to bind the intact spike. And this fits with other papers we've seen in the literature where if they sort with the S2 antigen alone, you get a higher frequency than if you look for cells that bind both S2 and intact spike. So we think that's a characteristic of some of the antigens, and it seems to depend a lot more on different preparations or different sources of the S2 antigen that you use if you try to use this as a domain by itself, even if you have an extra trimerization domain added to it. So that's just a bit of sort of inside caveats to think about with this, but it applies to lots of other things like flu hemagglutinins. Some of them, if you buy them commercially, they're really not intact trimers, and it's sort of a caveat emptor situation there. Like, watch out for the quality of the antigens. But anyway, we then, okay, fine, we're looking at all this DNA tag information. How well does it really reflect the specificity of the B cells? So here are the DNA tag counts in blue. Now we have the actual B cells, or here we've made monoclonal antibodies from a bunch of them. Each of those is a column. The DNA tag count from the original B cell is in blue for the different antigens. This is the binding that we saw in ELISA or MSD format assay for the monoclonal antibody made from the BCR sequence we got from each cell. So they're pretty nicely correlated. There are certain ones that we see, for example, that had both nucleocapsid and spike antigen binding. These ones are probably polyreactive ones that we shouldn't trust particularly. We did pick them up initially with more signal for the spike than the nucleocapsid. Doesn't mean that these might not be involved in the response to the spike, but we were a little less trusting of them. But overall, we thought that the correlations looked really nice, and we felt that, gave confidence we could trust the interpretations of the data based on the DNA tags alone, which is great because here this is about, I think 70 or so different monoclonals. It takes quite a while to make that many and test them all. This is the same thing with the antigen variants. So you can see that they show a similar correlation. Here we extended it to include Omicron variants that showed up later in the pandemic. And we also included the ACE2 blocking assay with the MSD format where you test whether the antibody can prevent ACE2 from binding to the RBD. So that's what these data show. So it was all pretty correlated, and a bunch of the antibodies that we found, they did actually have binding and even ACE2 blocking for Omicron variants as well. These ones were selected to be some of the more broad ones that we found. So the nice aspect is then you can look at, well, what are the combinations or what are the different sets of binding to different antigens in different combinations you can get out of these data. So here we looked at the infected versus the vaccinated to see whether we could see any sign of what we saw serologically, that there might be greater breadth in the vaccinated people compared to the infected, at least at the earlier time points. So here overall, the pattern had some correlation with that. Like the most common category of B cell in the vaccinees that we detected were ones that bound both the original Wuhan RBD here at the bottom, but also bound most of the variants that we had in the panel except for the beta. And you know, some other ones that bind several variants were also relatively common compared to those that just bound a single variant. Um, the um, the infected individuals, the most common category for them were ones that bound just the Wuhan RBD, and then they had some broader ones as next categories. So this is not a quantitative comparison here, however. Um, one other, yeah. So if we just summarize overall, like we make up a weighted variant breadth, so counting the more escape-prone variants with a higher score than the ones that are really similar to the Wuhan antigen, as a way to make this into a sort of a quantified um, score. We do see that if we look at, for example, the cluster 13 class switch B cells compared to the atypical ones, we see that there's actually lower breadth in the atypical B cells compared to that cluster 13, or the ones that look like they were recent germinal center emigres. When we look at the time course and look at the breadth that we detect in the binding, we see that it looks like there's some increase, which reaches significance by month 3 overall in the responses in the class switched cluster 13 group. Amongst the atypical memory B cells, the data were fairly sparse, but we basically did see that there wasn't as much of a change detected in the relative breadth for that specificity. So that's something that I think has, not been all that well explored in terms of different B cell populations and their antigen breadth in real human time course responses to vaccines or infection. Um, the other aspect is, well, what happened with the somatic mutation in the BCRs? In this case, it's from the heavy chains um, of the different B cell subpopulations that were specific during the time course. And so we could see that if we looked at the atypical memory B cells, their somatic mutation frequencies were not zero. I mean, they often had a few mutations. But when we compared it to the somatic mutation of the antigen-specific cells that were in that class-switched um, cluster that had, the IL-4 receptor and IL-21 and that sort of thing, over time they showed a, much more dramatic increase in the somatic mutation frequency of the heavy chain. So that was suggesting to us, these ones that look like they may have been in a germinal center, well, it certainly fits with the mutational data that we see. And this contrast, I think, I haven't seen in many other studies overall. So I think it is telling us that the biology of these atypical ones really is different, even though they're a part of the normal physiological response to lots of infections and vaccine responses sometimes as well. So Shanti Kotagiri, who I mentioned, has done some other modeling or analysis of these data, trying to combine all the other parameters that we have from the repertoires as well as clinical characteristics. So this way of showing the data, in each case, each column is a little cell, and here are annotations of the different kinds of other data we had from both the binding of the B cells to the antigens, or other things like, what was the tissue of origin. We had some data from deceased organ donors that passed away relatively early in the pandemic also. So we did some analysis of their specificities, for example, using the same experimental approach of sorting with those panels of the DNA tagged antigens. So what we see here, basically there seem to be some ways of classifying based on the different antigens that were bound. The B cells up here, the B cells here, these are the ones that bound most broadly to the many variants in the panel. Ones that were progressively binding fewer of the RBD variants are the ones that have less blue blocks filled in here. We looked for correlations with things like, well, what about the light chain type or the B cell phenotype, the isotype of the antibody, mutation level, other characteristics, and we put this into a more formal kind of model as I'll describe. But one thing that was pretty evident was that the BCR receptor composition in terms of gene segments showed a good correlation with variant binding breadth, or so at least showed some notable um, differences. So if we look at B cells that bound only the ones that were closest to the original Wuhan RBD, that's Wuhan alpha variant and 417N, compared to ones that were more broad in their binding progressively as we go to the left here. And we look at samples from the blood or also from the organ donors. The advantage of the organ donors is you have way more B cells to work with than a typical blood sample. So there's more cells represented here. But if we look at the V genes used in the heavy chains of the B cells that bound those combinations of the RBD antigens, we see the following patterns. So in the ones that are not very broad at all, there's certain genes that are used, a little more frequently. For example, 169, 434. Some of the ones that are common, but also seem to be part of this response. As we get to ones that have additional variants added on that they can bind, things don't change too much. Those are really a lot of the similar usage patterns. But once we start to get to ones that are broader, including these 484K B cells, we see that some other genes start to become more prominent, like B353. And that's really most notable once we start to add the gamma variant as well as some other ones like this, 551 shows up. I think in this crowd, I know there's aficionados who'd have favorite ones of these and things like 459 and so on. But there was really a nice correlation. We know that now, maps onto the epitopes that some of these antibodies bind that were defined by the many beautiful cryo-EM studies that were done during the pandemic. So overall, we see some patterns like V1-2, 169, 434 were relatively more commonly used in the low breadth clones. And then some other ones show up as being associated in general with greater breadth overall. So Shanti used a sparse partial least square discriminant analysis to try to pull out more quantitative analysis of this to define like what features of the repertoires or other aspects of the antibodies were associated with less breadth or greater breadth, relatively speaking. So, for example, if we look at the cell phenotype that the antibody came from, being in that recent germinal citra emigrae, at least putatively, cluster, that was associated with antibodies that had greater breadth than those that came from the typical memory B cells. The repertoire associations for the heavy chain V genes, as I mentioned, the 353 antibodies, as well as a few other ones, seem to be significant in terms of being associated with greater breadth overall. And others were associated with less breadth, 434 1-2. Similarly, we did get some IgA antibodies in these experiments, and they were associated with relatively less breadth than the IgGs, especially G1. That was the major type. If we look at other things, what differed between infection and vaccination, some of it shows, as that there's actually relatively greater contribution of these cluster 13 class switch ones in the vaccine response, and more of these ones that were like the activated B cells in the infection, as I showed. This is a really distinct population for them. The repertoire usage here, these are being analyzed independently, so it's not reflecting the association with each subset. The infection responses had relatively higher proportions of the 2-5 and 1-2 gene usage. The vaccine responses in the time points we looked at had a modest but greater usage of the 353 antibody types. So that's actually correlates with some of the breadth that we saw in the binding data, and potentially could explain the serological observation that was the starting point for all this. We also looked to see, like in bone marrow samples that we had for the organ donors, were there differences in the frequency of representation of the B cells that had the different phenotypes? Those clonotypes, did we find them in the bone marrow where we sequenced with bulk IgH repertoire sequencing? None of this, I think, reaches significance because of the sparsity of the number of overlapping clones, but it certainly seemed as though there may be in the infected individuals more B cells that had a higher frequency in the bone marrow that we could detect the clones. Those seem to come from different subsets. So here's one from an atypical B cell. These other ones are from the class switch set and other phenotypes. So this is a topic that we're diving into in more detail now. So that's our SARS-CoV-2 stuff, and I'll just briefly show that we've been extending similar approaches to influenza studies as part of some really nice collaborations with Scott Hensley's lab at UPenn and the Ward and Wilson labs for structural studies. These are the lab members who've done the flu stuff that I'm going to describe. So a similar experimental layout. multi-antigen panels applied to human samples from infection or vaccination. Here, this is all going to be regular seasonal flu vaccine um, from the last 3 years, basically. And for this, we used an interesting HA antigen panel. So this is the hemagglutinin, major receptor on the flu virus, which along with the neuraminidase is used to type different flu variants. And it determines binding to the B cells based on based to the host cells based on binding sialic acid. But we can also sort out specific B cells that bind to these HA antigens um, based on having mutants of them that have decreased sialic acid binding, and also by neuraminidase treating the B cells before the experiment. So here we used a broad panel. It has some of the ones that are characteristic ones that infect humans all the time, H1, H3, and influenza B. Um, it also has interesting ones like this H5 antigen, which is, in this case, it's from the previous bird flu outbreak, 2004, Vietnam variety from that. But we also included a bunch of HAs that are more associated with avian infections and haven't really, rarely or never been involved in human infection. These include some of these higher numbered ones, H4, for example. We also included some HAs that are found in bats, so H17 and 18. The purpose of this was we thought it would be interesting to look at the breadth of recognition of flu variants in human memory B cell populations much more broadly than most of the literature usually deals with, to see how much cross-reactivity there is and what sort of patterns we might find amongst these different antigens. Part of that could be also to evaluate, like, how much H5 binding memory B cell, content do people's memory repertoires have in the context of the growing concern about an H5 outbreak with especially the recent jump into cattle in the US. That's a great worry for a new pandemic that could be, hopefully not, but could have a greater mortality than SARS-CoV-2 did, for example. So here's some overview of data from that. These are basically from from vaccinated individuals as well as healthy blood donors who have memory B cells for flu. So in each case, you'll see each row is a cell. These are based on, I think, about 1,000 B cells also. And we see the binding patterns to different combinations. Here we've grouped the group 1 flu antigens. So they're all more similar to each other than they are to the group 2 flu antigens. That's just a branching in the phylogeny of the flu tree, basically, a phylogenetic tree. But what you can see is that the H1s that you'd expect people should have memory to, we found quite a few cells that bind. But there was also cross-reactivity with a number of other antigens that people really don't see in infections or vaccinations, or at least not often. Encouragingly, the flu B binders were mostly distinct from the flu A, and the group 2 antigens also showed more correlation with each other than they did with the group 1, which was what you'd expect. So there's a lot of interesting stuff about how you analyze the data, and Evelyn Georgiev's group and others have been teasing out, like, what are the best ways of identifying a positive binding signal. I think that there's some of the earlier papers use this center log ratio approach. We tried that initially, but we felt it was probably better just to do an analysis based on plotting the binding tag frequency counts as and modeling them as a negative binomial distribution, because most cells are negative for most of the antigens, and then taking a certain high proportion, high percentile of binders as the one that you're really confident are specific. So there's been a lot of method development in that area, but if we look at the antigens and what are the correlations for the B cells of binding different combinations of antigens, what we see is shown here basically with the two H1 variants that we looked at. The B cell binding to them was pretty nicely correlated. This is just based on Pearson correlations, I think, of the individual cell DNA tag counts. If we look at within the group 2 antigens, like these two different H3s, they're also nicely correlated. And in general, the group 1 antigens shown in blue compared to the group 2 antigens shown in red, you see that there are these blocks of greater correlation of binding that map along with group 1 versus group 2 identity overall. I put asterisks next to some of these because these are ones we later had them tested in collaboration with Andrew Ward's lab at Scripps. And we found that these are ones where they had a higher proportion of non-trimer HAs. So these are ones where there's probably some monomers that were involved in the binding. In some cases, they may have been predominantly monomers. So the binding for those we trust a little bit less. And as you can imagine, setting up this kind of experiment is a big pain because you have to have all these antigens in good shape all at the same time, all ready to go for the sorting. So there's quite a bit involved there. But some other little interesting features that came out of this are that if we look at H4, which should be a group 2 flu antigen, it actually shows a lot broader cross-binding to some of the group 1 antigens as well. You can see here for the H1s, here for other group 1s like these H 567 or 568 and 9. You also notice that there's some antigens missing from this table, which are, for example, H2, right? A really important antigen infected people from 57 to 68 approximately in terms of the year, birth years. And that one we couldn't use because that is too high a background binding. But we recently learned in the literature and from other colleagues who've been studying this that some of the flu HAs bind MHC class 2 in addition to sialic acid. So that's a pretty, important observation that probably explains the background binding that we saw for H2 and why it wasn't able to be included in this panel. Okay, so this looked pretty reassuring that we're actually getting, blocks of correlated binding that we'd expect based on the flu group antigen types. And here, this is just showing general patterns. So, a lot of the binders we detected were for group 1 influenza A. These are group 2 influenza A. The ones that bound both some examples of group 1 and group 2 were relatively fewer. Some of those were, as I mentioned, the unusual ones like the H4, for example, and many fewer cells that actually bind across group A, like influenza A, influenza B. So let's see. Yeah, this is in showing in more depth what were the patterns of antigen binding that we see for the different HA combinations. And here we're also showing the isotype of the B cell that expressed each of those BCRs. So some of the things that show up are, we saw in some of these data, surprisingly high frequency of binding to H9 flu, but that was mostly in B cells that had IgM as their isotype. Most of them were not class switched. And that's in contrast to, for example, the H1 California variant. This one had also stability that was not perfect. There was some non-trimer antigen there, but still we saw a higher proportion that are IgG. And for binding to flu B and the ones that were, binding the broader groups of group 1 antigens, ones that bind group 2, they all had a predominance of IgG, as you would expect from, adults' memory repertoires. But the interesting thing about this table is this thing has a super long tail, as you could imagine, right? All the different other combinations of various of these antigens. So for any particular one of interest, like, H5, we got quite a few cells that bound it in some combination with other antigens in the panel. So we're still digging through these data, especially Artem Mikalev in the lab is digging into that in a lot of detail. But we think it's a really unique way to get a deep look at what people's memory contains for flu and how it's represented. So for those aficionados for the BCRs, we've been evaluating based on binding to the different antigens in the panel, what are the characteristic V genes that show, a higher representation of the cells that have that binding activity. So here we found a bunch of things that map onto other studies in the literature, for example, like V1-18 is really seemingly related to binding group 2 antigens, especially H3. So you can really see it showing up. Here again, we've coded the group 2 ones as red, group 1 flus as blue. So just to make it easier to tell them apart. But you can see that there's certain characteristic V gene usage for some of the antigen binding. Notably, these are for B cells using kappa light chain. If we look at ones using lambda, the patterns are totally different, as you would expect and as the literature would anticipate. So if we look at the lambda, you can see it's quite a different spectrum of reactivities that have binding for each of those respectively. So we think that this is telling us something about the epitopes that are being bound, and that's something that we're now digging into in more detail in analyzing individual clones. Similarly, well, what about the cross-reactive B cells? What are the V genes that are used a lot in those? So these are ones that they had to bind at least one group 1 and one group 2 B cell. These are ones that were using kappa. And so there's certain V genes that are kind of overrepresented, especially 118, which is prominent in some of the ones that are in the literature already. For the lambda using B cells, a different pattern, 439 shows up, 551, some others. So I'll leave off here so there's some time for questions. But we think these antigen panels combined with single cell transcriptome analysis and PCR sequencing, really let us look at the monoclonal or the individual B cell clonal responses at a depth that we couldn't do previously, just due to expense and the laboriousness of those experiments. So we're applying this in a bunch of other areas now. And some of the main observations from our SARS-CoV-2 work were that we do seem to see different subsets of B cells stimulated in the time courses we followed. Some of this is correlated with the antigen binding activity, but there's also maybe some non-specific, or at least not specific to the antigens in our panel, activated B cells that are in the infection but not in vaccination. We saw notable differences in the atypical B cells compared to class switch memory ones that had markers of recent germinal center exposure in terms of their mutation increases and their binding breadth. This correlates with the repertoires of usage in terms of the binding patterns we see for particular antigens. In the flu data, people's memory has a lot of binding to HAs that we've never seen, probably because of shared epitopes. Some of that might be important for helping to predict what's going to be a more dangerous new variant to show up in the population, or ones that would have crossover potential from avian or pig or now cow origins. So I'll leave off there, but just to thank again the people in the lab who did the bulk of this work, and our many collaborators who've done an amazing, supportive work in this stuff, for the flu stuff, the Hensley lab, especially as part of a center of excellence based at UPenn. For our COVID work, our many collaborators, Kari Nadeau, Mark Davis, and Bali helped quite a bit. For the organ donor stuff, Klaus Niemann has been the key collaborator there. So, and really many thanks to the MSD for supporting our serological studies, and this ATOM group who really made some really nice antigens as part of this work. The funders include NIH from several grants, and then the Gates Foundation and others. We especially want to thank the participants who consented to take part in the research. So thank you. Happy to answer any questions.
Thanks, Scott. Fascinating talk. I'm sure there will be many questions. Maybe I can start with one question. So in the first part of your talk on SARS-CoV-2, there was one slide where you showed that there was greater variant binding breadth for the vaccinee B cells versus the infection individuals.
Yes.
Can you elaborate a bit more? Yes, this one. Why do you think that is? And yeah.
Yes. So so this is sort of supported more by the more quantitative analysis that followed later, but this is showing the frequency, with this sort of upset R plot, the frequency of B cells that had the binding patterns shown in the lower part. So for each antigen combination, basically it goes in decreasing order how common that pattern was. So, just at an initial eyeballing of it, it seemed to us that there were basically, the most common category in the vaccines was quite broad in its binding of multiple variants, except for the beta. And also many of the other categories that were high up in the frequency list were also ones that were quite broad. But this is something that, at an initial glance is not sufficient. So that's why we had to do the quantitative analysis that showed that there was, in fact, greater breadth for the vaccinees than the infected. The other aspect of this is that as we looked at later time points, the infected individual B cells showed relatively increasing breadth over time as well. So that's something that fit with the serology also, where the proportions of their polyclonal plasma antibodies that bound to each antigen improved or got closer to what we saw in the vaccinees the whole time. But this thing I thought was really interesting. Like, why should the antigen in the vaccine give a different response from the antigen when presented in the context of the viral infection? And there's reasons why it could. There's some sequence differences, right? There's stabilizing mutations, proline mutations in the encoded antigen in the mRNA vaccine. But there's also the many differences that come from being, expressed in human cells from the modified RNA source that's in the vaccine compared to the actual, virus RNA itself, for example.
Mm.
We don't think that those antigens are necessarily identical. Um, so yeah, there's a lot of things to investigate further there in terms of like
Yeah.
what are the actual proteins made? How complete is the translation with the modified RNA, for example, that sort of thing.
Mm.
Um, yeah, any other questions? Let's see.
Jamie has a question.
Hi, Jenny.
Hey, hi,
Hey, long time
Scott.
no see. Hi, how are you?
Yeah, I'm good, thank you. Yeah, so I wanted to ask you a bit, I haven't really kept up with it about the atypical B cells.
Hmm.
I mean, a little while back, it seemed to me that there was some kind of overlap between them and the age-associated B cells, and
Hmm.
also these B cells that are kind of dormant or inactivate, inhibited, and are unresponsive to a lot of things. So is that still what's going on with the cell population these days? And I think in
Yeah,
the group here is also Nina, and she might have something to say about that too. Anyway, I was just wondering if you could catch us up with that. What's the thinking now? I know
sure.
Iñaki Sanz was saying for a while that he thought they were just early plasma blasts, but I got a feeling maybe that's not true anymore.
Yeah, I think it's a complicated topic, but these cells with this kind of phenotype and different variations of it, like which transcription factors do they have at which levels, that sort of thing, have shown up in a number of different disease contexts. So, as you say, like in aging studies, but also in different autoimmunity studies, it's increasingly evident that they also are part of normal responses to vaccines and infection in a lot of cases that have been examined. So, I think that they're you know, what's really going on in the autoimmune disease case compared to healthy immunity? Is it just that if get B cells responses underway, you will have a certain fraction of that happen with the atypical phenotype cells compared to the regular class switch ones or non-class switch but non-atypical? That's something that's still under investigation, but I would love to hear Nina's
Well, and also,
take on
can I
that.
just throw in a chronic infection like HIV?
Yeah.
I think you see
Yep.
elevated. Yeah.
Yeah, right. There are these ties to like, is there energy of some kind going on, or are those B cells able to be stimulated further, or that sort of thing? I mean, they're clearly encoding good specific antibodies in the COVID stuff that we looked at within the timeframes that we're talking about. But yeah, Nina, what do you think?
Yeah, no, so it's a great question, Jamie. And the first answer I have for you is I really don't know. Even though we work on these cells, they're very confusing. And they're somewhat amorphous in terms of their description. Of course, their characteristic features are that they express a transcription factor TBET, and they actually have a fair number of other signaling molecules that would suggest that they are upregulated or enhanced in their activity by innate stimuli. So, TLR7 or TLR9 ligands. So things, which could come up in severe infection, for example, in COVID. So that was one of the theories that I and many other people had early in the pandemic, was that these atypical or age-associated B cells, or whatever you want to call them, that express TBET, are responding to an environment where you have a lot of cell damage, and you have an ability of these cells to kind of make an innate like response, kind of you have the Titanic, and you just kind of go all hands on deck to try to deal with the overwhelming infection, and you have a lot of cell injury. And in diseases like lupus, we presume similar things occur. So during disease flares, you can see significant upregulation of these types of subsets. And then there is also, Inaki Sans and others have shown that there is this differentiative pathway towards plasmablasts and antibody secreting cells. They're kind of transcriptionally and epigenetically poised to turn into those types of populations. So that's also interesting. And then ABCs, age-associated B cells, also have distinct anatomic distributions and contributions to protective immune responses to viruses as well, right? And so they can contribute a lot to antibodies that are protective to flu. So it doesn't surprise me at all that you see them in this kind of analysis. But how they versus germinal center B cells contribute to protective immunity, I think, is something that people are continuing to work on. Certainly Mike Cancro and others, I think, have shown that there is this differentiative pathway towards plasmablasts and antibody secreting cells. So that's also interesting. And then ABCs, age-associated B cells also have distinct anatomic distributions and contributions to viruses as well, right? And so they can contribute a lot to antibodies that are protective to flu. So it doesn't surprise me at all that you see them in this kind of analysis. But how they versus germinal center B cells contribute to protective immunity, I think, is something that people are continuing to work on. Certainly Mike Cancro and others have ideas about that as well.
So let me just point out too to you all that Iñaki, I guess he's had some surgery recently, but he's supposed to be putting together a workshop in terms of what do we do about nomenclature around these guys. And in fact, he's in charge of the IUIS B cell nomenclature, B cell and plasma cell nomenclature. And but the the argument or the criticism that I'm hearing is that in fact he's never actually, nobody's come out with the full nomenclature for B cells. So I don't know, I'm not really deep in that area, but I know a little bit about it. So I'm hoping that you guys might be able to get involved in that discussion if you're interested.
Oh, when I look at all those cell atlases, it seems like there's only 3 kinds, so it should be pretty easy, Jamie. There's like
Yeah, I don't know. I mean, we've been mapping these ABCs in tissues of human organ donors, and by sort of 10x platforms, there are several different subsets, we think. So,
Yeah.
but it's complicated. I think that's if Inaki wants to grab the bull by the horns and dictate to us how to name these things, I'm actually kind of fine with that at this point.
No, I think what he wants to do is have a
Yeah.
workshop and I know you're on the list, Nina, and Scott, I'm wondering about you. Yeah, no, and IUS has given him money to do it. So, so I think the idea is to get people together and have a little think tank.
that would be a good benefit to the field, I think. I don't want to steal time from Suhas's presentation though.
Yeah. Maybe we ask for 1 or 2 more questions for Scott if there are more questions. Maybe just one last question because we didn't talk so much about, I mean, I really think the data set that you have generated is amazing and we need more of these kind of data sets. So please keep generating these kind of antigen specific data sets. I was wondering because you didn't show a lot on the sequence similarity of those sequences. So are they very similar to one another? Are they diverse? And did you check also overlap with existing antigen-specific databases, especially on the SARS-CoV-2, where we have already this large database from the Charlotte Dean lab, right? So do many of your sequences fall in already known antigen-specific RBD known specificities, or is it completely new? Can you say something
Yeah,
about that?
sure. Yeah, just for real briefly there, we've done a lot of analysis about how many, what fraction of these sequences may be convergent or public clonotypes. There's certainly some overlap with ones that are in the COVI-AbDab database, and for example, like the 353 using B cell clones, those were, a ton of those were sequenced in the context of neutralizing antibodies and that sort of thing. So a fraction of the ones are, but with the, with the less biased binding-based data set that we have, there's a lot that are not convergent within the current, COVID-AbDab, but that we know are public otherwise also, so that we see them in multiple people. We've compared this, of course, to like bulk repertoire sequencing in hundreds of individuals, so we get sort of a better idea of like what the publicness is of a bunch of these. So I think there's a lot that's not been captured because of the way that the analyses were done for a lot of the antibodies that became the focus of neutralizing antibody papers that are missing, or don't really capture the full scope of what people made. A bunch of them were also neutralizing antibodies, but just weren't the focus. So yeah, so there's a lot more there.
Great.
Thank you, everyone, for the questions, and just I love this whole community, and it's a real pleasure to be interacting with you guys.
Thanks so much, Scott. Really nice talk.